from lungs will go to the heart. I'll now request Dr. Dhandapany to come onto the stage, please. Dr. Dhandapany is a faculty at the Institute for Stem Cell Science and Regenerative Medicine in Bangalore, and he was associated with Oregon Health and Science University in Portland as well. He joined inStem in 2016 as an assistant professor. He is interested in identifying novel genes and curative drugs for cardiomyopathies using a multimodal approach involving various modal organisms. Over to you, Dr. Dhandapany.
Thank you, sir, for inviting me for this talk. So for today's talk, I'm going to talk about the functional genomics of children's heart disease, especially hypertrophic cardiomyopathy. Hypertrophic cardiomyopathies are a group of heterogeneous heart muscle disease leading to thickening of the heart muscle and ultimately leading to diastolic and systolic dysfunction. As shown here, compared to the healthy individuals, these individuals show increase in intraventricular septal thickness, which is a hallmark of a hypertrophic cardiomyopathy. 1 out of every 500 people are affected with this condition, and the causes are mainly due to sarcomeric encoding genes. So worldwide, there are many attempts to study the other causes, which include 142 genes now. However, the South Asian populations are not explored promptly. So the focus of my lab is to identify new genes, mechanism, and therapeutic targets for hypertrophic cardiomyopathy, thereby advancing the precision medicine. The approach that we take is we collect large number of the patient family samples and perform next generation sequencing to identify novel genes, and once we identify novel genes, we actually use the cellular models to know the preliminary or important function, and here in this case stem cell models, and once we have relevancy in the functions, we go for a larger cohort to establish specificity and frequencies, and once we have a good number of frequency, we actually do some of the animal models to understand the mechanisms and therapeutic targets. So based on the proposed work, actually I divide my team into genetics team, mechanistic team, and therapeutic team. So for today's talk, I'm going to talk about the work that my postdoc did, Anupam, in collaboration with Ankit and Vignesh. So unlike adult cardiomyopathies, children's cardiomyopathies are life-threatening cardiac abnormalities and poorly studied. And approximately 70% of the cases are with no known cause, and there is no therapy for these conditions. So most of the children are associated with syndromic form, especially with cardiomyopathies, and one such syndrome is Noonan syndrome, discovered by 1965 by Jacqueline Noonan, and these kids are usually short in stature with webbed neck, pulmonic stenosis, and they have a high propensity of cardiomyopathies, and they do have a juvenile maloperative disorders, and approximately 1 out of every 2,000 live births are affected with this condition. So in order to discover new genes for that, we actually collected samples from South India, North India, and North America from Laurie Weiss lab, and we used these samples as a discovery phase samples, and for the replication phase we collected samples from Japan, from Rumiko Oshi's lab, and Marco Tattaglia from Italian lab, and Bruce from East Coast, and the numbers are given here in the parentheses. So approximately we have 60 cases, the total number of cases in the initial discovery phase, along with the respective familial controls. So we did a whole exome sequencing in that, and we used in-house variant filtering methods, and finally we got 16 missense mutations, and we filtered them using Gonome AD and recently released Asian-specific Genome Asia 1000K and Clinivar. And finally, we also filtered using recently a hosted data set by us, which is called Indian Database for Healthy Aging Individuals. And these individuals are healthy aging individuals above the age of 80 with no known risk factors, especially with the top 5 disease-killing genes. So parallelly, we also analyzed network analysis using the already known genes in order to prioritize the candidate genes. So we use cluster 1, ENCODE, and GILT by association, and we combined these things and made an RO algorithm, and there also we saw PRKCA as a candidate genes. So So we identified actually 6 genes, and one of the genes is PRKCA, which I'm going to talk to you. So shown here is the gene mutation or amino acid changes that we observed in the Indian families. As you can see, in the first family, there is a change at the 149 pushed into serine to asparagine, and in the second patients, there is a change from aspartic acid to 207 to glycine, and the third patients of valine 560 to leucine. And the genotype-phenotypic correlation revealed that they all are young, and as I said, the first family is of familial nature, the other 2 are de novo in origin, and they have a prominent phenotype which include short stature, in some cases mild hypertrophy, and in other cases predominant hypertrophic cardiomyopathy, and in one case it also have a valvular defects. So we could able to replicate this gene in the Japanese families. As you can see, there are 2 de novo families actually. So one of the mutation R216 is actually, now we are able to replicate the Indian cohort also. So the other mutation, the V566L, is also present in the Indian cohort. So the genotype-phenotypic correlation revealed that they are young and they do have hypertrophy, and interestingly, both the cases have septal defects. And so far, all the mutations what we identified are evolutionary conserved and predicted to be pathological using various in silico analysis. However, we find no mutations so far in the Caucasian population, either the patient from the Lowry's lab or Bruce lab or Marco Tartaglia's lab, suggesting that it might be a South and Southeast Asian specific mutations. So what is PKC doing in the heart? So PKC or PRKCA codes for a PKC alpha protein, and shown here is a diagrammatic representation of the protein along with the amino acid changes. As you can see, most of these amino acids are located in the calcium binding domain, and there is one in the regulatory domain, and there is one in the kinase domain. So the functions of the PKC or paleothera, especially they are involved in metabolism, proliferation, apoptosis in the heart. And they mainly signal to RAF1 AKT ERK pathway, and they importantly phosphorylate troponin I, thereby establishing a contact with the sarcomeric protein genes so that it helps in the contractile process. So we superimpose these mutations on the already known model, crystal models. So as you can see, these mutations are far away from the ATP binding site, suggesting they are allosteric in nature. So we then measure the kinase activity. As you can see, the kinase activity of the mutants are high compared to the wild type, but exceptionally one mutation didn't show kinase activity. Then we actually treated them with H2O2. The reason to treat with the H2O2 is since the cysteine that is located in the protein actually is a redox sensing molecule over there. So because of that, we treated with H2O2, and we see an upregulation of kinase. So we then express this mutation in the neonatal rat cardiomyocyte, and expressing the mutation in the neonatal rat cardiomyocyte shows increase in cell size and sarcomeric organization compared to the wild type, and this quantification is shown here. And they also show increase in hypertrophic marker, especially ANP, because this hypertrophic marker is actually a marker that re-expresses when the heart is under stress. It is a fetal gene marker, so we can see an upregulation in ANP, suggesting indeed the PKC mutants induce cardiac hypertrophy in a cellular model. And since we know that the PKC signals through RAF and ERK AKT, we actually measured the AKT and phosphorylation of ERK as shown in the plot. The phosphorylation of AKT and ERK are upregulated compared to the wild type control, suggesting indeed the PRKC mutant hyperactivate MAP kinase. So in order to know the precise mechanism, so what we did is long story short, we actually identify a particular serine site on the RAF, which is here at serine 499 region, through which the PKC imparts its function. So when we actually make this mutation towards makes the amino acid towards alanine, and we can by abrogating this function, we can able to abrogate the hyperphosphorylation of AKT and ERK, and this suggests indeed the PRKC mutants depend on the RAF serine 499 region. Next, in order to know the human relevancy, we actually generated the human stem cells model. The first what we did is we took the human embryonic stem cells and we expressed the wild type along with the mutants and differentiated them into the cardiac lineage and stained them for the cardiac markers here in the straight troponin I and smooth muscle cells. And this figure shows that indeed the cells that we obtain from the ES cells differentiation is cardiac cells, where we stain for troponin T and CAP-C, which are the cardiac specific markers. And these cells, after differentiation, they do beat and they have a difference in the contractility. So parallelly, we also obtain a couple of representative patient fibroblasts and convert them into induced pluripotent stem cells and then made a cardiac lineage. And here in this case, we selected using MLC positive cells, which is a cardiac specific marker. So for this particular experiment for differentiating the cardiac cells, we actually first block the GSK3, thereby activate the Wnt signaling, and after 4 days we block the Wnt signaling and thereby making a cardiac lineage cells. So at the day 14, and these cells quite nicely beat, and now we are measuring the the contractility basically, but I'm not going to show that. Whereas what we did is we actually stained those cells. As you can see, the staining of the cells basically a mutant shows increase in the cell size and sarcomeric organization compared to the wild type, and we can able to recapitulate all the cellular finding because where we saw there is an upregulation of the phosphorylation of AKT and there is an upregulation of the phosphorylation of ERK, and indeed many of the mutants except the 149 shows phosphorylations of the troponin I, suggesting indeed PKC mutant can induce cardiac hypertrophy. So in order to know that this happens in an in vivo model, we actually generated a Drosophila model as shown here, expressing these mutants in the representative mutants ubiquitously. The Drosophila actually shows a short stature like the kind of the patient that we observe, and also expressing these mutants using the GMR specific driver that expresses the eye actually shows increase in R7 receptors and ultimately showing a rough eye phenotype, suggesting they are a gain of function compared to the wild type. Express these mutant proteins in the thorax using a UBX3 driver, and you can see that the thorax region, the segments become shortened, and also expressing the mutations in the heart cells actually shows increase in sarcomeric organization, and all this data suggests indeed the mutations can affect the skeletal and heart muscles. And as we do know that it signals through AKT MEK pathway, we actually express these mutants in the Drosophila I and cross them with the AKT RNAi and MEK RNAi flies. As shown here, the AKT RNA flies and MEK RNA flies are able to rescue the phenotype, suggesting indeed these two pathways are crucial for imparting its function. Next, we use this Drosophila model for a drug screening purpose. For a drug screening purpose, we actually modeled a pupil lethality model where we express this Drosophila, mutated protein in the Drosophila heart and induce pupil lethality and mix the drug in the food, and drug efficacy is given as a simple curve as shown here. So if the drug is able to rescue more than 25%, we designate that as a kind of a moderate drug, and if it is rescuing more than 50%, then it is a kind of efficient drug. If it is able to rescue more than 75%, and we designate that as a high efficient drug. In this way, we actually took 150 clinically relevant kinase inhibitor, and we found in a preliminary studies that many of these inhibitors are actually able to rescue, especially that falls into the pathway of the RAF-MAP kinase and the P13 kinase, able to rescue the phenotype. However, the MEK inhibitor shows considerably high efficient, approximately 70%. So, in order to check this drug in a more relevant preclinical model, we actually generated the mouse model, and so the mouse model actually it's the overexpression of the particular mutant in the mouse heart, as the overexpression of the mutant in the particular, in the mouse heart actually shows increase in cells, heart size and increase in septal width thickness, and the M-mode analysis using ultrasound image actually shows the left ventricular or the posterior wall thickness is increased in the mutant compared to the wild type controls. And we also are able to replicate whatever we found in the cellular model, that increase in the AKT and ERK phosphorylation in the 4-month-old mice. And we also observed that increase in hypertrophic markers here in this case, which include ANP and SIFT in the alpha MHC to beta MHC and also alpha skeletal actin. So these data indeed suggest that the PRKCA mice can induce hypertrophic cardiomyopathy. So in summary, PKC is a novel gene for hypertrophic cardiomyopathy in syndromic cases, and Drosophila stem cell mouse model able to recapitulate the human Noonan syndrome hypertrophic cardiomyopathy phenotypes. AKT and ERK pathways plays a crucial role in this process, and MEK inhibitor shows an efficient rescue in the fly model. Now we are checking that in the mouse model. Now it can be used as a clinical diagnostic purpose. So with this I stop, and I thank all my Indian collaborators, especially the clinicians, and in my international collaborators including UCSF, NIH, Harvard, Mount Sinai, Tokyo University and Stanford collaborators, and inStem NCBS facilities. And my funding comes from inStem core funding, American Heart Association, Oregon Health and Science University, Wellcome Trust, and SERB DST.
Thank you very much.
Let's stop here and take questions.